Welcome to the Imaging One World talk today. We're very lucky to have Viji. Now pronouncing your surname is something I should have asked beforehand. I'm not even going to try. Draviam. Who leads the Centre for Cell Dynamics at Queen Mary in University in London, and she did a PhD in Cambridge with John Pines and on type B cyclins, and was a very enthusiastic PhD student, and is still very enthusiastic. I have very fond memories of her in the Gurdon Institute, and has had an excellent career looking at all sorts of kind of like, especially microscopy. Her microscopy has been quite inspirational, and hopefully we're going to see some amazing images today. And it's probably better if she introduces herself and talks rather than me. So I'll hand over.
Thank you, Alex. And yes, as Alex put it correctly, I think I spent a lot of hours in the underground of Gurdon, where all their fancy microscopes used to be. And Alex was managing them and making sure that we all are well trained and the microscopes are in like excellent shape. So I was kind of spoiled, let's say, during my PhD days. So I fell in love with microscopy during my PhD, and I'm still in love. So I'm going to present today what my lab does from a methods point of view, because then I can really dwell into what we have developed as microscopy methods to allow us to see cell division. When I say see, directly observe cell division at a resolution we've not been able to see earlier. Okay, so I'm going to try, there you go. So this movie, when it runs, you will see a plate of cells undergoing cell division. This is just one space, and we usually visit points just like everybody else does. And we acquire information from hundreds of dividing cells, information in terms of information through time, information through space, to understand how cells orchestrate cell division. So the reason we are interested in this area of science is because cell division defects are at the heart of aggressive cancers, fetal abortions, many types of intellectual disabilities, and premature aging. You know, that's something I start thinking about as we age, as I age. But it's interesting how errors in cell division underpin so many diseases. And that's because cells divide in our body throughout our life. Every single day, thousands of our blood cells undergo cell division, and they have to be replenished on a daily basis. So to understand cell division, what happens at the subcellular level has to be understood. So what you saw in this movie in green is basically DNA. It's a histone GFP marker. So you were visualizing DNA, which condenses into chromosomes, lines up into a plate just before the time when microtubules separated into two equal sets. And this process has kept me, I have to say, it's magical. If you look under the microscope, watching cells divide, it's absolutely magical. The process is orchestrated by microtubules. And I want to show you a video of how dynamic microtubules are. So microtubules are polymers, as many of you would know. These are formed of subunits of tubulin. Tubulin, when added, results in the growth of microtubules, and tubulin, when it's removed, results in the shrinkage of microtubules. And anything that's tethered to the very tip of the microtubule, you can imagine, can be pulled or pushed depending on whether the microtubule is shrinking or growing, respectively. But the question here is to understand, how does the microtubule know precisely when it should push or pull, and how much should it push or pull, because those are the decision-making events that ultimately ensure that our DNA is segregated into 2 equal halves during every cell division. Right? Understanding this requires imaging across scales. So what do I mean by imaging across scales? We have to image and look at the process at the level of these polymers, which are barely 30 nanometer wide structures. And we have to also ask, how do these polymers come together to build a spindle, which is in tens of microns in size? So you can see there are 3 orders of magnitude difference, and we have to image across those spatial scales. There is another important scale that I want to emphasize is the temporal scale. So microtubules grow and shrink in the range of microseconds or milliseconds, whereas the spindle itself moves and pulls and pushes chromosomes in the order of seconds. So there is a scale out there in terms of time as well. These 2 structures behave very differently, right? Now that means for microscopists like us, it's a complex problem of not only optimizing for that temporal resolution, but also we have to optimize for the spatial resolution. And I want to throw one more complexity into it, which is, if we move forward, the spindle. So what you see here is the mitotic spindle. It's capturing these chromosomes at a very small spot, a platform. I mean, in here it's a spot, but it's a platform made up of over 100 different proteins coming together to assemble a platform called the kinetochore. So in this image, what you are seeing them are those red spots. So what my lab and what my research has been all about is, what is the composition of that platform? How does that platform help in capturing chromosomes, capturing microtubules, so that chromosomes and microtubules can be attached? And what happens if there are errors in the process? Now, that's not what my talk is going to be about, although it's an important part of basic biology, where we aim to understand how microtubules ensure the accurate division of human cells. And we take from cells one step forward to understand the process in also 3-dimensional organoid models. So these are very small cysts of Keikutu cells. They can form these 3-dimensional structure in which, when you are studying mitosis, you can actually also think about how is the mitotic spindle itself oriented, not just about how microtubules capture chromosomes. And that allows you to look at the impact of mitotic defects in multicellular organoids. So these are exceptionally good preclinical models. Now, both of them have this beautiful way of being studied through microscopy. You could study it using high resolution. You can also look at it at high speed. As I told you, if you want to look at very rapid processes, then we need high speed microscopy. There is FRAB, FRET. The standard microscopy procedures can all be put into studying cell division. And the data set that we get helps us build models. And to test our models, we will obviously want to study it in extensive, large scale, where automated image analysis is the only way to move forward. So these are the tool developments that I would be really focusing mostly on today. I'll also touch upon how this has helped us understand the biochemical composition, which I told you already a little bit about, what these red spots, the kinetochores, are made of, and how microtubulins capture these kinetochores. And the long-term goal or the real impact is when we can take all this information at the molecular level and information at the cellular level for drug discovery. So today's talk, I'm going to divide it into 3 parts so that it gives me an opportunity to touch upon all that I have just told you. So I'll take you through what is the challenge in imaging photosensitive cells like the mitotic cells. Where does the challenge arise from? How we manage that challenge. And I'll also tell you another challenge where we are just beginning to develop tools to make sure that all the image analysis and movie analysis is not just manually done, because that's how it's done in most systems. And I mean, my own group also prefers doing that, but that doesn't allow high throughput scalable analysis. So I'll also tell you how we are using artificial intelligence to develop tools that will help us automate the analysis process. And the third one, third challenge actually brings us closer to the long-standing challenge, which is the imaging across scales challenge. How do you look at each and every microtubule end of this entire mitotic spindle? So you want high resolution and high speed imaging. How do we achieve that? So I'll introduce you to liquid lenses and how in my group we are starting to use liquid lenses for 3D images, imaging in the millisecond scale. Okay, so overall you will see that we do some microscopy and then we need some computational tools, and then we go back to microscopy and computational tools. So methodologically, that's what is happening in these problems. But as I told you, this is just the methods part of the lab. Our lab is mainly driven by questions that are central to understanding how is mitosis controlled spatially and temporally. So I'm going to give you a small crop of a movie to showcase where does photosensitivity arise from in the system. So in green, what you are seeing is the nucleus stained with histone GFP. So it's not really stained. Histone GFP is a green fluorescent protein fused to histone. So every nucleosome in this DNA technically should have histone fused to GFP, illuminating the entire nucleus. And this is an interphase nucleus. And you will see the DNA undergoes over 1,000-fold condensation to make sausage-like structures, which are called chromosomes, as you know. And these chromosomes would line up along a plate, but the cell will wait for that very last chromosome to line up, and only then it will undergo segregation. So I'll run that movie just once more so that you can see the arrow marking that one chromosome, which randomly is not yet part of the plate, and the cell waits for that, and then it undergoes segregation. So each and every cell will capture its chromosomes differently and align its chromosomes at different points. So we want to know not only how are cells capturing these chromosomes, but also how are cells monitoring the status of capture? How do they know that that last chromosome has aligned or not? Because if the cell prematurely started anaphase, then one of the daughters will have one chromosome more than the other, which is the starting point for meiotic defects, which lead to Down syndrome or Klinefelter syndrome. So, many developmental defects arise from errors in this process of chromosome microtubule capture. So dividing cells, as you just saw, are highly dynamic, and they are highly photosensitive because that when we are trying to image this process, the DNA has gone through condensation, so it's all tightly wrapped up and not really amenable to the DNA repair machinery. So any amount of DNA toxicity that it experiences will either kill the cell right away and will just commit apoptosis, or would just not move forward through mitosis in a normal way, giving you basically no meaningful data. So studying dividing cells required specialized live cell imaging conditions, especially if you're sure that you want to capture reliable dynamic information from 3D stacks. And even when you have the specialized imaging setup, we have to accept that it's always going to be discontinuous long-term movies. And that's when I mean discontinuous, what I mean by that is all conventional microscopes are built in a way that the machine would step up in microns to build your Z stack. And when you're in the topmost Z stack, you're obviously not having the information about what's happening at the bottom Z stack, right? So that's what I mean as discontinuous. So the information that we get is not continuous. And then even if you are imaging at the faster speed, if you wanted long-term movies, you probably would space it up. So temporally, it would also have these gaps. So these are the two challenges that are faced by everybody who studies cell division or any dynamic subcellular process. So we wanted to think about how can we overcome this. And today I'm going to tell you 2 or 3 tricks that we did, which hasn't allowed us to fully overcome, but we can overcome it substantially. So the first thing that we did was we thought of integrating 2 different cameras to, I mean, we use Deltavision. So this part of the talk is focused on Deltavision microscopes. But what it allowed us to do is to visualize the process at 2 different resolution or 2 different, for 2 different fluorophobes at high speed. So this is something we developed at that time. Deltavision was with Applied Precision. So we developed it with Applied Precision and IMSOL. And we could actually visualize for the very first time. So this is in 2012, 2013. We could visualize for the very first time how these kinetochores, you remember the spot-like structures on chromosomes, how they are captured. So we could show that the microtubule ends don't capture kinetochores directly, but in fact, these kinetochores just bind to the walls of microtubule fibers, and we termed it lateral attachments, and how these laterally tethered kinetochores would ultimately become end-on tethered kinetochores and remain attached to the end. At this point, it was somewhat a surprise because it had been shown in yeast that such a process would be possible in Saccharomyces cerevisiae, but we could also observe it in human cells. And I'll run this movie and you will see why this is not trivial to capture this. So you're watching the spindle trying to capture these spots, but while you're watching, you can see that the spots are appearing and disappearing because they're just moving through Z. Right? So this is what I meant as there is discontinuity in information, and we need to acquire a lot of Z stack. So my very first PhD student with whom I worked quite a bit on this because this problem was really close to my heart, he won the Cambridge Sense Prize because this setup really allowed us to track these particles through time, which was not possible before. But I want to show in the next slide a summary of what spawned from that, right? Because once this method was set up, we could really use RNAi tools, mutant tools to dissect various steps of the process. We could show that the very first step was the kinetochore, which is the structure that builds at the center of the chromosome, just tethers onto the microtubules in whatever orientation it can. And after tethering, there are kinesin-like proteins that drag the chromosome all along the microtubule to the very tip of the microtubule. But in addition to the kinetochore being moved towards the microtubule end, there's also the microtubule end that's shrinking and appearing closer and closer to the kinetochore, the microtubule attachment site. We could really figure out what are all the key regulators of this process. And the molecules are all written in yellow here. And we could show that once the kinetochore arrives at the very tip of the microtubule, then there are proteins that are recognizing the state of attachment. This is the Astern-SCAP complex, and maintaining the state of attachment by bringing the appropriate enzymes, in this case, PP1 phosphatase. Now, all of this could be dissected because we were able to visualize this process and track the events through space and time. Now, as you can imagine, by the time a lab looks at so many mutants, so many RNAi-mediated depletions, protein depletions, and hours of movies, there's just terabytes and terabytes of 3D movies, and these are high quality movies because you can actually look at each and every kinetochore fiber and how it captures the kinetochore. But classifying these types of attachments, the movements, the steps, so this was like really time consuming, tedious, sometimes can even introduce errors. But more importantly, I was worried that by manual analysis, we might be omitting some subtle but reproducible changes that might explain some significant phenotypes, right? So that's one advantage of automated analysis. If we could analyze something through a machine, you could just run the analysis several times, increase the terabyte of data set. It wouldn't be bored. It hopefully will make consistent errors, not errors that are inconsistent. And it can save the time of really creative lab members. So one of the things that I wanted to really do was to somehow develop tools to track objects. So the first thing we started tracking, let's take the easiest one, which is the bulky object, and try to track this. And for this, the example that I'm going to choose is spindle. So spindle, although is bulky and then a very large structure, the complexity with spindle tracking is the spindle is a dynamically changing object. So even within the same movie, the spindle would either grow or shrink, tumble, change in different orientations. So let's say a spindle looks like this in X and Y with the chromosomes on the metaphase plate very clearly. Through time, the spindle can move in any orientation, and you have information that is changing through time and space at multiple channels. So you can have the cell membrane being tracked in a label-free channel, the chromosome being tracked, and then the tubulin being tracked. And so this is somehow relatively very easy for the human eye to put it all together. You know, it looks very simple. Okay, there's a cell, and then there's a spindle, and then a chromosome. But you throw this information to a computer for automated analysis, it would just choke because it cannot cope with the discontinuity of information. So it cannot really connect. This spindle is exactly the same spindle that I show on the right. Sorry, I don't know whether we can see my laser pointer. I'm going to just try. Okay. Functional, there you go. Okay, so a computational tool would not be able to identify that the spindle at time point 1 is precisely the same structure at time point n. Whereas a human eye, even an undergraduate student who has never seen a spindle in his or her life, you show them, they will know these two are the same objects, just turned a little bit around, right? So this increasing complexity in image data analysis is what is choking automated image analysis. So while my group was at Cambridge, we attacked this problem by computational vision tools. So telling the computer that, well, what you see in red is actually the spindle. It will look like an ellipsoid structure of size X, Y, and Z, and go and use standard image segmentation tools. It did work. It enabled automated tracking, but we needed someone who constantly worked with those movies to, pick those spots up when the computer failed and said, I can't figure out the continuity here. Then the student would come in and change. So Adam Corrigan, now he's at AstraZeneca, he wrote all the code for spindle 3D based image segmentation based spindle tracking. And Ehsan Zulkifli, she did all the screening and tracking of the spindles when the computer just failed. We did manage to do a huge screen, and I have to say Adam and Ehsan worked very hard to track it together. And we did learn new mechanisms that are regulating spindle movements, but I knew this is not scalable. This is not scalable because although the machine is doing most of the tasks, it is still recommending the user to come and double check or meet the gaps that it cannot meet. And when we went back into these movies at areas where the computer could not connect, it was really trivial two positions. It's just that the machine perceives these two positions as diametrically opposite or different, but it's for the human eye, we could just meet this meet this gap. So the limitations was it needed manual adjustments. So if a spindle was just 10% small, you had to immediately tell that to the computer. It could capture, you know, big changes in spindles, but when it was small changes, it struggled. It could not really say whether that was a phenotype or there was a problem in imaging. And it could not be used for precise 3D measurements, because we realized that although the image segmentation tools, you know, we used very standard ones, could recognize where the spindle structure was, because as you can see here in the binary image, it's very, very clear where the spindle begins and ends. It quite could not say whether the spindle was tilted in Z or it was perfectly in XY. So it could not connect the structure in 3D. So we needed something else. So more recently, well, 2017 feels a very long time back, but relatively recently, we started looking at machine learning tools. So machine learning tools, although are very exciting, they were very powerful and trying to solve image analysis process for automated driverless cars. So we thought, why don't we use that for tracking spindles? But they needed a lot of work in the front of cleaning the image, labeling the image, augmentation, and training and building the model. And finally, the building the operation part is just a small part of it. But we took this approach, and David Dang, a joint student with the Shastri group, decided to go for a deep learning architecture. So we used the KNN network, and we chose this because it's dynamics blind and it's easy to implement. It can be a supervised machine learning approach, and it's very good for classification and regression type of problems. So as you can see here, it can identify where the spindle is very well, and also where the chromosomes are, and it could do it through different stacks. But what we needed to do still was bring that continuity in. So we added some mathematical modeling steps to connect those gaps between the 3D slices. And this made sure that it's now dynamics aware. So it connects not only in Z, but also through time. So we can now begin to see where the spindle's principal axis is and how is the axis moving relative to the cortex. This was very, you know, although we started in 2017, I have to say, I just loaded this paper 3 days ago. It took us a very, very long time to build a clean deep learning method with modeling tools for tracking spindle dynamics. It's on bioRxiv if you want to see, but for the purpose of the talk, David made a very nice movie for illustrating this point. So what you are seeing is SpinX tracking the spindle poles through time. So pole 1 is in yellow and pole 2 is in black. And you can see as the poles move and search across the cortex, you can actually observe the movement through time, the reduction and change in pole to cortex distance. And we are able to see this now, partly because we already had these high resolution movies that helped us build the software, but also because the key part is the mathematical modeling, which allowed us to say how the spindle poles should be connected in a mitotic spindle. So we are now working with Zeiss to transfer this knowledge into their image analysis online platform called APIR. So it will become very soon available for everyone who wants to look at dividing cells. So very quickly, I want to move on to the third challenge. So we are able to look at the entire spindle. At the same time, I told you there is also the spindle microtubules that build the spindle that are being regulated, which ultimately affects spindle function. So how are these microtubule ends regulated? What we already know is that the very tip of the microtubule has EB1 end binding protein 1, EB3 end binding protein 3 complexes, which go and bind to the very tip of the microtubule, and they regulate the growth of the microtubule. So in this movie, these dots that you see are basically the tips of the microtubule. So I hope I can run this movie. So these tips can be tracked in 2D, but they cannot be tracked in 3D. Now, this again is largely because of the continuity problem. They can't be tracked in 3D simply because when you are imaging in one plane, your comet, the tip of the microtubule end, might have moved to another plane. So EB1 tracking within spindles have been a long-standing gap. And we wanted to solve this without having to buy very expensive microscopes and set up high-speed 3D imaging. So Dr. Noriko Hiroi, she's a visiting postdoc from Keio University, and she brought with her, she used to work in the liquid lens field, and she brought with her this opportunity to use electrically tunable liquid lens for 3D imaging. What are liquid lenses? So these are electrically tunable lenses. They have a liquid polymer, so they allow continuous focusability. You can change the curvature by simply adjusting the power, so you can change focal length within milliseconds. But that also means it introduces errors in terms of how the objectives allow light paths. So we have a review in JCS just telling about all the difficulties with using electrically tunable lenses. So obviously they are not as good as our glass objectives that have very standard and specific substrate which do not change in refractive index or curvature. But ETLs have been successfully used for wide field microscopy, 2-photon microscopy, but it was not yet set for deconvolution microscopy, because deconvolution microscopy, as you can imagine, relies on that PSF being perfect so that you can use it for deconvolving the objects. So what we did with Noriko, and this is something that Kuniko Minolta was interested in because they were interested in for using it in their own cameras, was to develop methods to allow us to correct for changes that would appear during high-speed imaging and changes that are introduced by this liquid surface, the liquid substrate that electrically tunable lens are made in. So here I just put a picture of the electrical tunable lens so that it's very easy to see. So it's just kept here at the bottom of the objective. Of course, it introduces a lot of aberrations because you're now suddenly changing the length and position of your objective. However, you are able to correct this. If you are able to correct this computationally, then we don't have to worry about it. So that is one of the goals. So we are just beginning to build this, and what I show here is a sweep of a 3D image sweep. So basically within milliseconds, you can just go down and come up with a sweep, and you can see how quickly you can connect these particles through time. Chris is a new PhD student in the group, and now he is trying to set it up in a way that we can generally adapt it to any wide field microscope. And a wide field microscope that's capable of using deconvolution algorithms. So where do we think these kind of tools can be applied? So the use of these kind of tools is where we need high throughput imaging, but also high speed imaging. So I'm going to spend like a couple of minutes telling you about a pharmacogenomics project in the lab where we really could see the benefit of this kind of technology. So you probably have heard of the connectivity map. What that simply means is that we take transcriptional signatures from either individuals, models, cells, and ask how transcripts increase or decrease after being exposed to a particular drug. And then you treat the same model with many, many different types of drugs. So thousands of drugs. And then based on these transcriptional changes, you can reposition these drugs as being closer in terms of how they affect the biological system. So the connectivity map is a large project which aims to use gene expression signatures so that it can connect small molecules, genes, and diseases. So we could look at this connectivity map and identify new microtubule stabilizers or destabilizers that look just like standard microtubule altering drugs that we use in the lab. So for example, are there new drugs that look like Taxol? Or there are drugs that look like Nocodazole? So you could easily query this data set to ask which of these drugs might be closer to, not structurally, but closer in terms of how it affects the transcriptome. Once we had a bunch of drugs of that kind, we wanted to just throw them on cells and watch microtubule behavior. So you can imagine now you will have hundreds of drugs at hundreds of different dilutions and combinations. You want to apply them to your cells and track microtubule behavior. Now this problem becomes just not manually, it cannot be manually parsed. That's where you need automated tools for microtubule end tracking. So this is a collaboration with the Wellcome Sanger Institute, where Francesco Iario helped us identify signature that looks like paclitaxel, and we could pull out a bunch of drugs that would give the same transcriptomal change as paclitaxel would do. And we could line these drugs up as newly repositioned drugs. So this is a computational prediction. And then we had to bring those drugs back to the lab and try to understand whether the density of comets, so here the comets refers to the EB1 and EB3 comets at the very tips of growing microtubules, we could ask whether the density of comets were changing in presence of these drugs. So here is a very good example for how advances, even very small advances that we make in microscopy methods, can make a large change in high throughput image analysis. So, to just conclude this study, what we could show is that splitomycin and glipizide, these are two drugs that are not yet known to affect microtubule dynamics, but we could see in the lab that adding splitomycin or glipizide within a few seconds would alter the growth and shrinkage of microtubules in epithelial cells. So to summarize what I have told you today, I opened my talk with interest in imaging mitosis at high spatial and temporal resolution. And I explained to you how adding just a dual mount for a camera helped us look at a process at very great detail. And the process that I told you today, we termed it end-on conversion because it allows us to monitor how microtubules capture kinetochores on the lateral wall and convert them into an end-on state. I also introduced you to a deep learning method that we have developed to automatically track spindles if you are interested in the software tool or knowing more about the methodology, please feel free to catch me, because now we are at the stage of transferring the technology to Zeiss for enriching their API environment, and it will become commercially available as well for tracking spindles in 3D. I also, so that's, and what I didn't have time to take you through is how SpinX has helped us understand spindle positioning mechanisms. We have been able to identify new kinases that sit at the cell cortex and regulate the movement of spindle by pushing the spindle. So classically, the spindle is thought to be moved by pulling forces mainly in human cells, but by looking at hundreds of spindles undergoing thousands of time points of movements, we could say categorically that spindles are not only pulled, they are also pushed, and we could identify the kinase that's responsible for this regulating the pushing movement. The last one that I told you is about a microtubule drug repositioning study where tracking submicron objects, that is the EB1, EB3 comets, helped us dynamically reposition drugs that look like paclitaxel. And the electrically tunable lens for this particular tracking is just under development. So what we are trying to do is focusing on emerging data analysis challenges. So let's say we have the electrical tunable lens that allows us to track multiple particles. How can we connect to gradients? So particles are very defined objects. Can we connect that to gradients in the cortex to explain how dynamic changes occur? So Chris is doing the study at the moment. We are also interested in tracking nanostructures in the super resolution regime. So all the study that I've shown you so far are using deconvolution microscopy or confocal microscopy or using electrically tunable lenses. But we are also interested in understanding structural changes of these objects in using super resolution microscopy, because we can actually guide this analysis through electron microscopy data. So basically connecting that gap between super resolution microscopy and electron microscopy at the spatial level. And the end-on conversion process is really fantastic for that, because the kinetochores are moving from laterally attached state to an end-on attached state. And during the process, this large submicron structure undergoes dramatic changes in its structure, and we want to be able to visualize this. At the moment, Catalina, Sinong, and Sanji are all focused on understanding how kinetochore changes are driven in cells. We are also looking for a postdoc. So if you know of someone, and if you are someone who is interested in developing AI methods, it's a great time to be in the lab, because we are developing tools to grow organoids and generalize SpinX for multicellular studies. So we have an open postdoc position for anyone who's interested in developing AI methods for microscopy. Now, before I close the talk, I also wanted to highlight two things. So in Queen Mary, beyond the Draviam lab, there are many single cell imaging labs. And this is a small advertisement for all those labs in the Center for Cell Dynamics. And we will also be hosting the first ELIRA super resolution microscope for photokinetic studies. So we can really look at dynamic protein recruitment and release from different parts of the cell. That's all the people in the lab. I'm really grateful for these fantastic young people who come and bravely take all the complex microscopy problems, and thanks to all the funding bodies who have supported our research. And thank you for your attention.
That's great. Thank you very much, Viji. I feel there should be a round of applause there, but possibly virtually rather than. So if anyone has any questions, then please put them into the chat, and then we'll ask you to ask them in a few moments. We're going to have the quiz, which Nick is going to be the master of ceremonies for, just to see whether people were paying attention. And it's supposed to be a hard challenge this week.
Yeah, there's a definite mixture of cell and technique questions in there. So I just put the link in the chat there. So if you click on that, you can participate to win a fold scope. So actually, I don't have one. I've never won one. I'm not allowed to participate, but anyway, it's a great thing to have, and it's a bit of fun. So please do try and join us for the quiz. And if you just stop sharing, I can share my own screen. Just a minute. There we go. Right, so I'll give you that. Let's go into present mode. Okay, so you should see the first slide of the competition. So while people are joining, hopefully, yeah, we've got a participant. The cell division is amazing. There you go. There's already one comment there. Let's see if we can get a few more other people joining in. A couple of messages. All right. A few more. Let's wait a little bit longer so we can get some more people in. Otherwise, it's going to be a three-horse race. Go on. I know there's a lot of people out there listening to this talk, so let's see if we get a few more people joining in. Okay, that's better. It's less. Let's wait one more minute. Trouble is, when I put this quiz up, I can't actually see what's going on in Zoom anymore. So let's see. We've got 7 participants, 8. All right, I'm just going to wait a little bit more, and then 9. Oh, there we go. If we get to 10, I'll get it started. All right. Okay. Let's go with that one. Okay, and here are our participants. Let's see. Okay, here's the first question coming right up now. What is the end-on conversion process? Chromosomes are captured at microtubule walls and brought to the microtubule ends, or chromosomes are directly captured at microtubule ends, or capture of microtubule ends by chromosomes. Which one of these is the correct answer? And right, 4 good answers there. Right, excellent. So next, let's see what happens next. There we go. Let's see who's answered first. Looks like Earthman is the winner so far. Hannah and Kathy quickly behind. Okay, let's see what happens next. Next question. There we go. Which of the protein complexes below have been shown to play a role in microcephaly and chromosome segregation? That's cyclin B and CDK1, SEN-P and A, or astrin and SKAP. Let's see who is paying attention here. The correct answer is astrin and SKAP. All right. So let's see where we are now. Puffy zooms into the lead. Wow. Okay. Oh no, sorry. MDM is in the lead. All right, Hannah's a close second. Let's see what happens next. This is good. We're getting a bit of a change in the leaderboard here. So let's see, question 3. Deconvolution microscopy aids one of these below. So which is the best one of these? It aids sampling with a pinhole to reduce artifacts. It works with a measured PSF to give increased resolution along the Z-axis or it can be used for deblurring to improve quantitation. And the best answer, yep, is number 5, gives increased resolution in the z-axis. So let's see where that takes us now. MDM still in the lead, but Micah's crept into 2nd place. Okay, right, there's still all to play for. Hannah and Puffer, you can definitely catch up. What is the marker for the growing plus ends of a microtubule? EB1, EB3 proteins, MCAK and kinesin, or NDC80 and NUF2 complex, which is the marker for the plus ends. Now I know the answer to that one. I was definitely listening at this point. There we go. Okay. EB1, EB3 proteins. Right. Let's see where that puts us in the leaderboard. Wow, Micah zoomed into the lead. Okay. Let's see.
Excellent. Thanks for that. So I guess.
Oh no, we're not
Micah...
quite finished. Got one more.
Oh, are we? Oh, there's another question.
5 questions today. This is it. How does a liquid lens enable high-speed axial focusing? By rapidly changing the amount of liquid in the lens, by rapidly moving the axial position of the lens, or by changing the electrical signal applied to alter the focal length, curvature. Everyone, 5 answers there. Okay, right, here we go. This is the leaderboard here. Let's see who came out on top. Micah, you are the winner. If you could just make yourself known to the organisers, you will get the grand prize of a Foldscope. Okay, great. Thank you very much all for participating, and I will at this point stop sharing. Okay. Thank you very much for that.
Oh, thank you for that. That was quite a hard quiz. Knowing the answers, I still managed to get them all wrong. So, so on that, we should ask Micah if you're still there. Micah. So you had a question. Is a frame rate 10 Hertz or 0.1 Hertz per stack?
Can you hear me?
Yep.
Okay, well, I'm just confused because it just said that there was a frame rate was 10 seconds. I didn't realize it's like is the frame rate like 10 seconds per stack, or is it 10 frames per second? 10 stacks per second. So which one? I think it is the first one. So it takes 10 seconds to acquire a stack, right?
Yeah, it's the first one. So we tried to keep video rate. It's the first one. So we have to think slightly differently from how we look at conventional imaging system, in the sense, usually we will think of it as, is it 10 slices for this distance? But here the control is not in terms of slices. So the lens is continuously changing its focal length, and the camera cuts out at some point. So that's how you get like a still, not really still, semi-still image.
Okay, and you also had a follow-on question as well, I believe.
Yeah, so it's a bit provocative question, because I know Deltavision is based on a normal illumination, so regular UV lamp, I guess, or LED, depending on. And then, am I correct?
Yeah, it's a wide field microscope. Yeah,
yes.
But I mean, you could have lasers, but if you think, I just saw your question. It's a very good question. Why don't we use confocal? So the reason is really photosensitivity. So we want to make sure that we get all the 3D information as much as we can when we have illuminated our cell.
So you are aware that the laser might be phototoxic more than a normal lamp?
Yeah. For a long period, I mean, not long period, even now, Laser-based sectioning is not the best way to go for mitotic cells at that stage, because the toxicity is very high. That's if you want long-term imaging. If you just want short-term imaging, then yes, these days you can get very gentle lasers, but that's because the cameras have also become way better. Let's say the recording systems are way better, but still, if you wanted long-term imaging, the best way to do is to just go through wide field and then deconvolve the data or use computational tools to get the information out later on.
Thank you very much. This answer made my day.
Thank you.
Excellent. So does anyone else have any questions for Viji? So I guess, so on the electronic tunable lenses, so have you tried looking at deconvolution and setting up different kind of like variables for different adjustments of the lens, or?
That's what we are starting, Alex. That's really what we are starting, but it's not, I mean, it's not so easy, because you have to get the lens, I mean, the drivers to smoothly talk with the microscope, you know, driving software, and they all are kind of closed. So we're trying to use Micromanager to somehow reach the microscope controller. So we have just started.
Oh, okay. No, I'd be interested to see how you get on. I guess because you'll change the lenses, you say go deeper, or the situation changes. So it'll be an interesting thing to see.
That's why you're right. You're right. I'm also very, very excited to know what will happen. I put it as a challenge 3 because I think we are going to not get the answer like in a year or 2. It's probably a long problem.
Yeah, no, I can imagine. I can imagine. So, and Nick has a question.
Yeah, I just wanted to ask you about
locally, but the free version is cloud-based.
Okay, right. So you're sort of basically putting your algorithm out there, and then we can sort of send our data over to it. Okay.
Yeah, so if for non-commercial use, so you know, researchers like us who are kind of poor people, it's free. But drug development industry, so if they wanted to do pharmacogenomic screens, repositioning, the idea is that they should be able to pay.
Right.
Seems reasonable. So any other questions from anyone? No? Excellent. No, that was a really brilliant talk today. Thank you very much, Viji. That was,
thank you.
Yeah, excellent. And I look forward to rewatching this on YouTube at some time in the future. And trying to get the answers right to the quiz. So
I hope you edited that part off.
So anyway, thank you very much. And it's a rather embarrassing time where everyone kind of says, kind of put a reaction or something. You can't virtually clap. So very well. So, but no, brilliant.
Thank you.
Thanks very much.
Bye.
Thank you very much.